welcome to this explainer. So, have you ever looked in the mirror and felt that despite whatever your actual chronological age is, parts of you might be aging faster or maybe even slower than the rest? Well, it turns out that intuition is absolutely correct. Our bodies actually possess these incredibly mysterious, hidden internal clocks, and they definitely do not tick at the same speed. Today, we're unpacking a truly groundbreaking 2023 study published in Nature by a team of Stanford researchers. I'm telling you, this paper completely revolutionized how we understand human aging by proving that we can actually read these individual clocks.
Picture this. Your body actually has a high-tech dashboard, kind of like what you'd see in a modern car. But instead of just a generic check engine light, this biological dashboard displays real-time, high-fidelity data on 11 critical organ systems. We're talking your heart, your brain, your kidneys, the whole shebang. And the wild part? The Stanford researchers discovered how to read this dashboard not by doing invasive biopsies, but through plasma proteomics, which is just a fancy way of saying they study the proteins floating right there in your blood.
To get a full grasp on this, here is our roadmap for the explainer. First up, we're decoding organ-specific aging. Second, we'll explore blood as a data bus. Third, calculating the age gap. Fourth, predicting Alzheimer's years early. And finally, building the future biological dashboard.
Okay, let's jump right into part 1, decoding organ-specific aging. We really need to start by challenging the status quo of how we currently measure getting older. For decades, the medical field has basically relied on these systemic, blended scores to tell us our biological age. But honestly, an average biological age, what's often called a phenoage, creates a totally dangerous clinical blind spot. It's essentially a systemic blur. It masks a situation where your overall health score might look perfectly fine on paper, but a specific, absolutely critical organ, like your kidney or your brain, is silently and rapidly failing in the background. The Stanford study essentially proves that aging is not some systemic constant. It's actually a collection of entirely independent trajectories. Moving right along to section 2, blood as a data bus. To fix that massive clinical blind spot we just talked about, scientists needed a new way to listen to these individual organs without using a scalpel. This brings us to the ultimate information highway in the human body. You see, blood plasma isn't just a transport fluid for oxygen and nutrients. It is literally a continuously updating telemetry bus. Imagine a river carrying distinct messages from every single town it passes through. Your blood carries a proteomic archive of around 5,000 trackable protein expressions. As every single organ ages, or functions, or degrades, it releases its own specific proteins directly into the bloodstream. The data is all right there, just floating inside your veins. So, how do they turn that into an actual functional methodology? Well, the big challenge isn't getting the blood, it's making sense of those 5,000 different proteins. The researchers started with a foundational clinical baseline of 1,398 healthy individuals. They took blood draws and meticulously mapped out that massive 5,000 protein plasma array. Here is where the magic happens. Artificial intelligence steps in to act as a massive filter. Because when you have 5,000 sequence proteins, a ton of them are just general systemic noise, stuff related to overall body inflammation or general decay. So they use machine learning algorithms to ruthlessly filter the dataset, isolating only the proteins that are expressed 4 times higher in specific organs. By eliminating all that systemic overlap, they finally extracted 11 distinct, highly targeted biological aging clocks, which perfectly sets up section 3, calculating the age gap. Now that we actually have these isolated organ clocks, the next logical question is, how do we measure if an organ is actually in danger? We use a super powerful metric called the age gap, or HG for short. It's an incredibly simple but profound concept. HG is basically just the quantifiable difference, the delta, between your expected chronological age baseline and the actual biological age of a specific organ, based entirely on its protein telemetry.
When things are going well, you have a healthy pattern where your chronological age and your biological age match up beautifully. But if that gap deviates sharply upward by more than 1 or 2 standard deviations, it's a massive red flag. It signals severe accelerated aging for that specific independent organ. So we aren't just talking about a general feeling of getting older here. This is localized accelerated decay. And when you evaluate that age gap across all 11 organs, completely distinct patient profiles or phenotypes start to emerge. Some people are at a perfectly healthy baseline. Others show uniform systemic aging where everything degrades together. But the real stealth risk is when a person's average overall age looks totally fine, completely hiding the fact that there is a massive age gap spike in one specific organ. That spike reveals a single organ crisis long before any clinical symptoms would ever show up in a standard doctor's visit. Now for Section 4, predicting Alzheimer's years early. The theoretical science is definitely fascinating, but let's apply this powerful new metric to one of the most terrifying medical mysteries of our time. For a really long time, traditional clinical diagnostics for cognitive decline have relied heavily on the tau hypothesis, basically looking for plasma tau indicators. But the huge limitation there is that tau usually only spikes after irreversible cognitive decline has already started. The Stanford data flips this completely on its head, revealing a hidden combinatorial risk. It turns out Alzheimer's isn't just a brain disease. It is a highly complex intersection of localized brain aging and structural vascular or arterial degradation. And this right here is where the findings become truly groundbreaking. The researchers found that when both the brain age gap and the arterial age gap exceed 2 standard deviations, this model predicts the onset of Alzheimer's disease with an 80% certainty. And get this, it does this up to a full 5 years before any clinical symptoms appear. 5 years of preventative lead time before irreversible damage takes hold. That is literally unprecedented. Finally, let's wrap things up with Section 5, the future biological dashboard. Having 5 years of preventative lead time for a disease like Alzheimer's completely moves medicine from reactive to proactive. But how do we get this out of the Stanford academic labs and into the real world? Well, the massive barrier right now is the price tag. Mapping a comprehensive array of over 5,000 proteins for a single patient costs upwards of $15,000. So unless you've got a Silicon Valley billionaire's healthcare budget, it's totally inaccessible for routine clinical use. But don't worry, there's a brilliant workaround. By leveraging machine learning, researchers can identify the absolute highest weighted, most specific proteins for each organ clock. This means we can distill that massive, insanely expensive 5,000 protein array down into a highly targeted commercial panel of just 50 to 100 high-value proteins. We're talking about 5 to 10 key biomarkers per organ. That distillation slashes the cost exponentially, creating the exact foundation needed for an affordable, highly scalable predictive diagnostic platform. So, what this all really boils down to is a fundamental paradigm shift in human medicine. By achieving high-resolution telemetry, we gain the ultimate prize, preventative lead time. We don't have to wait passively for diseases to manifest and symptoms to ruin lives anymore. We can actively read the body's system telemetry to engineer early intervention. It leaves us with a pretty profound final thought about our own health data. This technology is rapidly maturing from expensive academic theory into targeted, affordable diagnostic panels. So if the tools exist to look 5 years into your biological future and pinpoint an exact organ crisis before it even begins, are you ready to see your dashboard? Thanks so much for joining me on this explainer, and I'll catch you on the next deep dive.